Thank you for the invitation to speak today. My name is Eiring, and I will be talking about a role for proteasome 26S subunit non-ATPase 3 in disease progression and drug resistance of myeloid leukemia. So my name is Eiring, and I'm an assistant professor in the Department of Molecular and Translational Medicine at Texas Tech University Health Sciences Center in El Paso. And it is early in the morning here for us.
Yeah, I'm so sorry for that, but yeah.
Okay, so just most people don't know where El Paso is located, so I just wanted you all to know that we are on the very far west side of Texas. Our house is located about 20 minutes from the border crossing to Mexico and about 10 minutes from New Mexico. So it's really more like New Mexico than it is like Texas. This is the quintessential star on our Franklin Mountains that lights up every single night. We have beautiful sunrises, we occasionally get snow, and we have spectacular sunsets. So I'm going to switch gears and talk a little bit about blood cancers. So in the United States alone in 2022, there's approximately 185,000 new cases of blood cancer expected to be diagnosed, which will result in roughly 60,000 deaths. And leukemia accounts for about 33% of all of those blood cancers, but 41% of those deaths. And leukemia is classified based on the type of white blood cell that's involved, either myeloid or lymphoid, and the rate of growth of those cells. The fast growing cells are considered to be acute. So we have acute lymphoblastic leukemia affecting the lymphoid population and acute myeloid leukemia affecting the myeloid population. And then the slower growing cells are called chronic, and that includes chronic myeloid leukemia and chronic lymphocytic leukemia. And I'm going to be focusing today on myeloid leukemias, both chronic and acute. So chronic myeloid leukemia is caused by the BCR-ABL oncoprotein, and this is a product of a genomic translocation between the long arms of chromosomes 9 and 22. And this is known as the Philadelphia chromosome. It's considered a stem cell derived but progenitor driven disease. And it results from the constitutive tyrosine kinase activity that's imparted upon the BCR-ABL oncoprotein because of this translocation. Now, this disease is highly treatable with tyrosine kinase inhibitors that target the BCR-ABL oncoprotein. However, resistance remains a problem, and particularly the leukemic stem cell population is primarily resistant to TKIs, and they remain after treatment and are a source of cells for future relapse. So we used an shRNA library screen to identify the genes that were critical for TKI resistance in CML. And specifically, I'm talking about kinase independent resistance. So one of the first mechanisms of resistance that was discovered in CML was BCR-ABL kinase domain mutations. But roughly half of patients do not have any explanatory kinase domain mutations, and they are considered to have kinase independent resistance. And this includes the leukemic stem cell population. And so we use this shRNA library screen to publish a role for nucleocytoplasmic transport as being critical to survival in TKI resistance. But there were many other genes that we noticed in the library. Importantly, 5 members of the Ubiquitin Proteasome system were hits in this shRNA library screen, including PSMA1, UBE1, PSMD3, PSMD1, and NED8. And the criteria for selection of these hits was a fold change of the barcodes for each shRNA had to be greater than 2-fold in our TKI resistant K562 cells versus the wild type parental TKI sensitive controls. And then each gene had to be targeted by at least 2 or more shRNAs targeting the same gene. And today I'm going to be focusing on PSMD3 and PSMD1 in myeloid leukemias. And so we performed RNA sequencing on TKI resistant versus TKI sensitive CML cell lines. And these cells were found to be enriched for TNF alpha signaling via NF-kappa B. And if you look on the right, many of the genes indicated in red are putative targets of the NF-kappa B signaling pathway. And when we looked at data in our RNA sequencing study versus published data in CML patient samples, we see that both PSMD1 and PSMD3 are upregulated in scenarios of TKI resistance. So I'm sure you all are very familiar with the Ubiquitin Proteasome system. It's a highly regulated multi-enzyme complex that maintains protein homeostasis. It's highly activated in diseases like cancer. It contains a 20S core catalytic particle that is responsible for the actual catalytic degradation of peptide targets, and then 19S regulatory particles that are responsible for substrate recognition and binding. And as you know, in diseases like cancer, the proteasome is targetable with proteasome inhibitors, particularly bortezomib, carfilzomib, and ixazomib. But like TKI therapy and CML, resistance and toxicity remain a significant clinical problem. So the 19S regulatory particle contains PSMD1 and 3, which is the topic of my presentation today. So the standard proteasome inhibitors target the 20S core catalytic particle, but some have hypothesized that targeting the 19S regulatory particle could inhibit the proteasome with less toxicity. So we confirmed in TKI resistant CML cell lines and patient samples that NF-kappa B signaling is upregulated in TKI resistance, in addition to nuclear phospho-NF-kappa B. And this was consistent in cell lines and patient samples. Now, PSMD3 in particular has already been shown to play a role in breast cancer, where it was shown to stabilize human epidermal receptor growth factor 2 from degradation. And it's also interesting to note that PSMD3 is just upstream of colony stimulating factor 3 gene, and mutations in this genomic region have been previously associated with changes in neutrophil count. So this suggested a potential role in myeloid leukemia. So our data shows that both PSMD1 and 3 are upregulated at the RNA level in patients who have progressed to what we call the blast phase of CML. And blast phase of CML is very similar to acute myeloid leukemia in that you have an accumulation of undifferentiated blasts, you have reduced responses to therapy, and patients often only survive 3 to 6 months after diagnosis. However, when we looked at the protein level, PSMD3 and not PSMD1 was found upregulated in CML patient samples versus normal cord blood controls. When we used shRNAs from the library screen to knock down PSMD1 and 3, we saw a reduction in growth of the TKI resistant cells to a much greater extent than the TKI sensitive controls, suggesting that these proteins are important for TKI resistance. And when we knock down either PSMD1 or 3, we see a reduction of colonies in both TKI sensitive and resistant cell lines, which correlated with the induction of apoptosis. We also saw a reduction of colony formation in CML patient samples, but importantly, knockdown of either of these proteins had no effect on cord blood cells, suggesting that if we can get bone marrow specific targeting of these proteins, they may be a good target for therapy. So ultimately, we showed that PSMD1 and 3 regulated NF-kappa B expression and transcriptional activity in TKI-sensitive cells, but to a more extent in the TKI-resistant CML cells. We showed that IL-6 is upregulated in TKI-resistant cells, and that it activates STAT3, which further potentiates NF-kappa B transcriptional activity in the TKI-resistant scenario. And knockdown of PSMD3 reduced K562 tumor formation in a subcutaneous model of CML. And ultimately, we published these data last year in the American Cancer Society journal, or in the Nature journal Oncogene. So, okay, but what about other types of cancers? So we used data from UALCAN to show that PSMD3 is upregulated in multiple different cancers compared with normal tissue. And as you can see here, this PSMD3 is upregulated in 18 out of the 24 cancers that were available in the database. And at the protein level, PSMD3 is upregulated in all 5 of the cancers that were available in UALCAN. And high PSMD3 expression also correlated with worse outcomes in multiple different types of solid tumors, including lung adenocarcinoma, kidney chromophobe, the kidney renal papillary cell carcinoma, as well as skin cutaneous melanoma and uveal melanoma. And so we asked, okay, that's great about solid tumors. Well, what about AML? So when we looked at every subtype of AML, we saw that PSMD3, but not PSMD1 expression correlated with worse outcomes. However, as you can see on the right, the survival curve kind of tapers off. And so we sought to look at different subtypes of AML. So CML is caused by a single genomic abnormality, the BCR-ABL oncogene. But AML is very heterogeneous and caused by many different mutations and molecular abnormalities. So when we zoomed in on different subtypes, we decided to focus on FLT3 positive AML. And you can see that patients with FLT3 positive AML who have high levels of PSMD3 expression have a sharp reduction in overall survival. So what is FLT3? Well, similar to BCR-ABL, FLT3 is a constitutively active tyrosine kinase when it is mutated in AML. And similar to CML, it is also treatable with tyrosine kinase inhibitors that target FLT3. However, resistance is a problem, and up to 60% of patients could either have adverse events or drug resistance. So we used shRNAs to knock down PSMD3 in FLT3 positive AML cell lines, and we confirmed knockdown at the mRNA and protein level. And we saw, similar to what we observed in CML, that knockdown of PSMD3 impaired colony formation of FLT3 positive AML cells. However, in contrast with our data in CML, this did not correlate with an induction of apoptosis, either in MOLM13 cells or in MOLM14 cells. In MOLM14 cells, you can see that PSMD3 knockdown did induce apoptosis in the absence of FLT3 TKIs. This particular graph shows quizartinib, but it had no additive effect when added with TKIs. So this suggested to us that perhaps PSMD3 is playing a different role in FLT3 positive AML than it's playing in BCR-ABL positive CML. And when we do FLT3 positive AML cell line xenografts, we confirmed these data in vivo. So knockdown of PSMD3 improved the overall survival of immunocompromised mice that were injected with those cells and treated with doxycycline chow. Also, in contrast with our data in CML, knockdown of PSMD3 really had no effect on NF-kappa B luciferase reporter activity in either of the FLT3 positive cell lines that we analyzed, further suggesting a different role for PSMD3 in FLT3 positive AML. So when we performed gene expression analysis, we looked at the genes that co-express with PSMD3 in AML, and we performed a pathway enrichment analysis. And that demonstrated that PSMD3 may play a role in energy metabolism in AML, particularly integration of energy metabolism and metabolism of RNA. So we used the Agilent Seahorse bioanalyzer and looked at functional metabolism in FLT3 positive AML cells. And as you can see, when we knocked down PSMD3, we saw a reduction of oxygen consumption rates in the knockdown cells compared with cells expressing a non-targeting control shRNA. So altogether, PSMD3 plays an oncogenic role in chronic myeloid leukemia by stabilizing the nuclear factor kappa B transcription factor. PSMD3 mRNA and protein expression are upregulated in multiple types of solid tumors, which correlates with worse outcomes. High PSMD3 mRNA expression also correlates with worse outcomes in FLT3 positive AML. PSMD3 may act as an oncogene in FLT3 positive AML by regulating energy metabolism, and we are currently looking at which fuel sources are really driving this phenotype. And then future studies will assess the toxicity profiles when targeting this protein in cancer therapy, and whether or not PSMD3 is targetable with small molecule inhibitors. So I have to thank my lab members at Texas Tech, particularly Josh Lara, who is a fourth-year medical student in my lab. I also need to thank a postdoc from my lab, Alfonso Bencomo, and two talented research technicians in the lab, Mayra Gonzalez and Idalia Olivas. This work was funded by a small foundation grant from the LCU Party Foundation, and also a Minority Medical Student Award from the American Society of Hematology to Joshua Lara, who performed most of these experiments. And that is all I have for you today, and I'd be happy to take any questions.
So yes, it is open for discussion. If anybody wants to ask, please come forward and ask. Dr. Anna